Imagine a room, not with soft blankets, lullabies, but with the gentle hum of advanced machinery. It's a nursery of the future. In the center, a newborn in a sterile, glowing pod. Holographic spirals of DNA float like digital fireflies. These are the building blocks of life, the very code that makes this tiny human unique. Scientists in crisp white coats watch data on transparent screens. They are not just checking the baby's heart rate. They are not just checking temperature. They are looking deep inside, at the instructions that built this child, ensuring every genetic letter is perfectly in place. It might sound like science fiction, but it's closer to our reality than you think. We are on the edge of a brand new world, where we can read the book of life and edit it. The technology is powerful, promising, and honestly, a little bit scary. So what is this amazing technology? It's called CRISPR. The name stands for clustered, regularly interspaced short palindromic repeats. But you don't need to remember that. Think of it like this. CRISPR is a pair of genetic scissors. Imagine your DNA is a very, very long book. This book contains all the instructions for building and running your body. It tells your eyes what color to be, how tall you should grow, how your heart should beat. It's the most important instruction manual you will ever have. And it's inside almost every cell in your body. Now, sometimes there's a typo in that instruction manual. This is a genetic mutation and can lead to serious diseases. This is where CRISPR comes in. Scientists can program it to find a very specific spot in the DNA book. Once CRISPR finds the target, the typo, it snips the DNA right at that spot. A precise, clean cut. After CRISPR makes its cut, the cell's natural repair machinery kicks in. Scientists can guide this repair, providing a correct piece of DNA as a template. The old instruction is gone, a new healthy one is put in its place, hopefully forever. This isn't just a futuristic theory, it's happening right now in labs around the world. Scientists are already using CRISPR to perform incredible feats of genetic healing. One promising area, inherited blood disorders. Take beta thalassemia, a tiny mutation in the gene that makes hemoglobin. Hemoglobin carries oxygen in red blood cells. The mutation means not enough healthy red blood cells, severe anemia, and organ damage, a lifetime of needing blood transfusions. Using CRISPR, scientists take stem cells from patients. They correct the genetic typo in the lab, then return the edited healthy cells to the patient. The results have been life-changing. In trials, patients once dependent on frequent, painful transfusions are now producing healthy red blood cells. They are essentially cured. A monumental achievement, rewriting our genetic code. Success isn't limited to one disease. Researchers are exploring many genetic conditions. Sickle cell anemia, genetic forms of blindness, muscular dystrophy, some types of cancer. The potential is vast. Even more amazingly, some researchers have gone further. Let's bring this out of the lab and into your life. Imagine you and your partner are planning to have a baby. You go through genetic screening. You discover you both carry a recessive gene for a serious condition like cystic fibrosis. This means there is a 1 in 4 chance. Your child could be born with this disease. It can cause severe lung problems, digestive problems. It shortens life expectancy. Now imagine your doctor presents an option. Through in vitro fertilization, they create an embryo. They can use CRISPR to correct that one faulty gene before implantation. Would you do it? Would you edit your baby's DNA to prevent a lifetime of suffering? For many parents, the answer might seem like an obvious yes. The instinct to protect your child is one of the most powerful human emotions. If you could spare your son or daughter from a painful, debilitating disease, it may feel like a moral obligation. This is the promise of gene editing, a chance to give every child a healthy start. But other questions start to creep in. Is this technology completely safe? The conversation gets even more complicated when we move beyond fixing clear-cut diseases. The same CRISPR technology that can correct the gene for beta thalassemia could, in theory, be used to alter other traits as well. What if parents could choose their child's eye color, hair color, height, athletic ability, increased muscle mass? This is where we cross a very important line, moving from medical treatment to human enhancement. We shift from fixing what is broken to designing what we desire. This is the world of designer babies, a concept that makes many people deeply uncomfortable. If this technology becomes available, who gets to decide which traits are desirable? It's easy to imagine a world where society starts to value certain characteristics over others. Would we see a new kind of social pressure, where parents feel obligated to give their children every possible genetic advantage? This could create a new and frightening gap in society, a divide between the genetically enhanced and the naturals. Access to this expensive technology would likely be limited to the wealthy, widening the gap between the haves and the have-nots on a fundamental biological level. The most troubling frontier is the idea of editing for complex traits like intelligence. Intelligence isn't controlled by a single gene, it's the result of a complex interplay between thousands of genes and environmental factors. Attempting to boost intelligence would be incredibly complex and unpredictable. What would be the unintended consequences of tinkering with the very genes that shape our minds? Could we accidentally alter personality, creativity, emotional capacity in the process? We are talking about changing the very essence of what makes someone who they are before they even have a chance to exist. This forces us to ask a fundamental question. What is the purpose of medicine? We are the first generation in human history to hold the power to direct our own evolution. The decisions we make in the coming years about technologies like CRISPR will have consequences that ripple through centuries. This is not a choice that can be left to scientists in a lab or to individual parents alone. This is a conversation for all of humanity. Should we use this power to change our own species? And if so, how? The fact that we can do something does not automatically mean that we should. The potential for good is immense, but the potential for misuse is equally staggering. The challenge is to find a way to embrace the incredible medical benefits of gene editing, while putting up strong guardrails to prevent its misuse. We need to create international agreements and regulations that draw a clear line between therapy and enhancement. This means fostering a global dialogue that includes scientists, doctors, ethicists, policymakers, the public. We need to decide, as a global community, what is permissible. Should we allow editing of human embryos at all? Or should we restrict CRISPR therapies to treating existing patients without making heritable changes? There are no easy answers here. Some will argue that we have a moral imperative to use any tool at our disposal to eliminate disease and suffering. Others will argue that editing the human germline is a line we should never cross, warning that we are stepping into the role of God. Ultimately, this technology forces us to look in the mirror and ask what we value most. The future is not yet written. We are all co-authors.